Thank you so much for the kind invitation, especially for Tony and Samir as well. It's a pleasure to be back here. For you, probably you don't know that I did my grad studies here in the UK at Loughborough, and it's always a pleasure to come back. So I'm going to talk to you today about antibodies targeting mucormycosis as another approach of therapy. I don't know why is nothing showing up here. I wanted to give you a brief introduction about mucormycosis. So it's basically an organism caused by organisms belonging to the order Mucorales. So that's what this slide is supposed to tell you. By far, organisms which belong to a family of the order Mucorales called Mucorales is responsible for mucormycosis is caused by Rhizospus, so which is approximately causes 70% of all causes of mucormycosis. Here we are. Just took a while. And then this is also true for, this is in the US, is 70% of all causes of mucormycosis. Leukemia and mucor are tied for second in the US, and I think also in Europe are tied for second as well, and you guys are still part of Europe, so I think this is true for the UK as well. We get more of an exotic cases of Syncephalestrum, Saxenia, and Cunningham as well. These are rare cases of mucormycosis. If you lump Rhizospus, leukemia, and mucor, probably this is responsible for approximately 90% of all causes of mucormycosis. So the disease itself usually happens in the immunosuppressed patients. Patients who are diabetics in ketoacidosis and other forms of acidosis are uniquely predisposed to infection. Hematologic malignancies, transplant patients, corticosteroids used. And there are actually immunocompetent patients that can develop mucormycosis, but these usually are subjected to severe trauma, such as soldiers during combat operations or during natural disasters as well. The problem of mucormycosis is the overall high mortality rates here, which is 50 to 100% based on the manifestation of the disease and the predisposing factors as well. Now, regardless of all kind of mucormycosis, all of them, they are characterized by the fact that they are extremely angioinvasive. So there is a lot of interaction happens between endothelial cells lining blood vessels and invading organisms. So one of the things that we were interested in understanding the pathogenesis of mucormycosis is trying to understand how the organism invade blood vessels. So Rick in my lab, basically what he did is he followed an affinity purification approach to understand what receptors, what mammalian receptors the organism binds to. So what he did is he took endothelial cell membranes and biotinylated them, and then incubated them with germ rings of different Mucorales. And you can see that there is basically this band which kept popping up. And by sequencing that band, it was basically determined to be glucose-regulated protein, molecular weight 78, which is a heat shock protein. It's mainly cytoplasmic protein, responsible for basically identifying misfolded proteins, and it initiates the degradation pathway for it. But also at the same time, being a heat shock protein, some of it basically gets relocalized on the cell surface during stressful conditions. And some organisms hijack that feature, bind to it, especially viruses, and basically invade the organism. So it just happens to be that Mucorales are really good at doing that as well. And this is really a unique feature in fungi among Mucorales. And you can see this is basically not only just confined to Rhizospus, but also mucor, Cunningham as well, they bind it. So luckily we have antibodies available against GRP78 from commercial sources. Rick basically incubated endothelial cells in the presence of Rhizospus and added this antibody, and he can show here that you totally abrogate the ability of the organism to invade endothelium in the presence of the antibodies, but not in the presence of a totally unrelated antibody. And there is no effect on adhesion here, so this is really an invasion. it seems like the receptor basically is specialized for invading organisms. And this assay also causes endothelial cell injury. So the process is you basically incubate the organism, it adheres to the endothelium, rolls over, invades, and then it causes injury. So if you block invasion, you eventually block injury, and you can see there is a 50% reduction in injury. He also followed another approach whereby he downregulated the expression of GRP78 in endothelial cells. These are all human umbilical vein endothelial cells, so they are actually primary cells, approximately 80% blockage. And these cells are resistant to invasion by rhizopus. And again, because of the fact that you block invasion, you block basically damage. Shortly after, George was interested in trying to identify what's the fungal ligand that binds to GRP78. So what he did is he did far western blotting by using GRP78 as a bait. And he identified a cell surface protein called COT-H. So COT-H stands for spore-coating proteins. So these are uniquely present in mucorales. You can't find them anywhere else, not in bacteria, not in viruses, not in mammalian cells, not in any genome that has been sequenced so far. Extremely unique. So if we've raised antibodies against COT-H, which are GPI-anchored proteins, you can see that in the presence of these antibodies, you completely abrogate the ability of rhizopus to invade the endothelium. And as a consequence, you also block the ability of the organism to cause injury to endothelial cells. George also basically downregulated the expression by RNA interference in rhizopus, and two different transformants basically were deficient in their ability to invade the endothelium. And as you would expect, they were basically also deficient in causing injury to endothelial cells. So just to emphasize the importance of this interaction between GRP78 and COT-H, we've basically stained either wild type or the mutants with attenuated expression of COT-H. And we've stained basically with GRP78. You see that it is really nicely expressed on the cell surface and accumulating around basically germlings that are in the process of invasion. And this is basically also staining COT-H in red. And when you overlay it, you see this nice yellow or light green color showing co-localization. So if you look at the mutant, you really see GRP78 expressed not to the same extent, and also it's really diffuse. It's really not accumulating around the invading organisms. And you can't really stain COT-H basically when you knock it out as well. And there is really no overlaying of the two colors. So COT-H is also important for virulence in an animal model. So these are mice infected intratracheally with rhizopus, whether it's wild type or those with attenuated expression of COT-H. And you can see that mice infected with the COT-H, they had 30% long-term survival versus 0% of mice infected with the wild type or the empty plasmid. And this is also corresponding with the fact that the mutant had approximately 1 to 2 log difference in target organs, lungs being the primary target organ, and the brain basically is a disseminated organ, so it's a secondary target organ. And the most amazing thing to me is here when you look at the histopath from these mice, the wild type and empty plasmid infected mice are riddled basically with Candida hyphae, and there is basically a lot of tissue edema, as you can see, whereas the ones basically infected with the mutant, you have more of a normal architecture of the lung, and you barely see any fungal elements as well. So we've done a lot of work on environmental factors present in certain patient populations who are uniquely predisposed to infection, namely diabetics in ketoacidosis who suffer from hyperglycemia, obviously, as well as elevated available serum iron and ketone bodies. BHB here, beta-hydroxybutyrate, being a representative. You can see in the presence of iron, you have overexpression of COT-H at physiological concentrations of 15 and 50 micromolar. Same thing with 4 and 8 milligram per ml of glucose, as well as for BHB. So these basically, in addition to this, there are the same effect on GRP78. So we've done a lot of work, and in the interest of time, I'm not going to be able to show you all the data about that because I want to talk to you about the antibodies themselves. But the model that we have basically that kind of actually explains why diabetics in ketoacidosis are uniquely predisposed to infection is you basically get acidification of the milieu by beta-hydroxybutyrate and other kind of acidosis. They compromise the ability of transfer intercalate ion because of the released protons. And that basically releases ion, higher concentrations of free iron, beta-hydroxybutyrate, as well as glucose, overexpress GRP78 on endothelial cells. They also overexpress COT-H on invading organisms, and as a result, you get basically enhanced invasion, tissue injury as well, and then you basically organ seeding. And at the same time, you have the detrimental effect of all these factors on phagocytes. So they really don't chemotax very well, and they don't kill the organism. So pretty much you're creating the perfect storm for this infection. So we've basically sequenced a lot of organisms from Mucorales, all clinical isolates, with the help of Vinnie Bruno at University of Maryland, Baltimore. And you can see all of them, they have COT-H, anywhere between 1 to 7 copies. So this is uniquely present in Mucorales, but it's universally present in Mucorales. And just to emphasize how invasive the function is, we've also added here basically Basidiolus and Candidiolus, and as you guys know, they usually cause superficial infection. They really don't cause any invasive infection, but they have absolutely no COT-H. So these are the first mold invasion that have been identified. Not only they are present, but they are also highly expressed. These are really highly expressed in spores as well as in hyphae. So one of the most expressed genes actually in Mucorales. With the help of Mike Heeman, we were able to create a model of COT-H, and the crystal structure of GRP78 is available. You can see that actually the COT-H3 binds really well to GRP78, and you see that this is really the binding faucet here. We were able to take a peptide, which was predicted to be antigenic, surface exposed, raise polyclonal antibodies against it, and we use these antibodies to see whether we can actually recognize Mucorales. Lynn in my lab, she can basically show that these antibodies recognize both spores as well as germlings, and they bind really avidly, as low as 3 micrograms per ml of COT-H basically shows the same amount of basically binding to 100 microgram. And also, this is not only just against Rhizospus, but also they recognize other Mucorales, as you would expect. This peptide was also common among all Mucorales, with approximately 70 to 78% amino acid identity. So all of them, they contain these COT-H. So buoyed by basically these results, what we wanted to do is we wanted to see whether these antibodies are protective. So what we did is we infected mice intratracheally. These are diabetic mice with slight ketoacidosis. And you can see that the mice that received basically a single dose of 30 microgram, which is really, really low amount of antibodies, are protected against the infection compared to those that receiving basically pre-immune IgG. These rabbit polyclonal antibodies. Again, this is also coinciding with the fact that there is reduction as well in the target organs, lungs, and brains. Same thing in neutropenic mice. So these are extremely protective in neutropenic mice as well to the same extent. The most important thing here is, here you go, yeah. So if you look at the histopath from mice that have been infected with Rhizospus, and these are neutropenic mice, and treated with either the isotype matching control antibody versus the anti-COT-H antibody, you can see there is a lot of hyphae with a lot of tissue edema and destruction in the lungs, whereas you see actually less basically tissue edema. There is some basically inflammation, but not a whole lot. The most interesting thing is, remember, these are intratracheally infected, so the primary target organ is the lung, but it disseminates to the brain. So you look here, you can see that there are fungal elements in the blood vessel in the brain, and actually some of it egressing outside the blood vessel and going and causing basically infection in the tissue parenchyma. You don't see that in the antibody-treated mice. So clearly those animals or the antibodies basically block invasion and progression of infection. In addition to that, we've taken basically tissues from these mice and we've determined the amount of myeloperoxidase as a marker for influx of neutrophils into the organs. These are diabetic ketoacidosis mice, and you can see that the ones treated with the antibodies, they have way higher concentrations of myeloperoxidase, indicating that probably these are probably cyanophagocytic and enhanced chemotaxis as well. We've taken neutrophils from normal mice versus diabetic mice, and we've basically treated them with either pre-immune serum versus the anti-CAT-H antibody, and we've determined the ability of these neutrophils to phagocyte rhizopus, and you can see that in the presence of the anti-CAT-H antibody, they are actually more phagocytic. The same thing with the diabetic as well. We've done lysosomal staining. I'm not quite sure if you can see it here, but basically there's a lot of lysosomal formation in neutrophils that have been incubated with rhizopus delamar and in the presence of CAT-H antibody here. This is also associated with the fact that these neutrophils were able to inhibit the growth of rhizopus as determined by the XTT assay, as well as the fact that they actually produce much better reactive superoxide against rhizopus as well. We know that these antibodies as well, they are cyanophagocytic through a mechanism that involves the Fc receptor. If you use antibodies against the Fc receptor, you totally block the ability of neutrophils from mice to inhibit the growth of rhizopus. And also if you use Fab fragments lacking the Fc, you basically have the same inhibition compared to pre-immune serum. So this is definitely an opsonophagocytic process through an Fc receptor mediated process. I mentioned earlier that this peptide is present in all mucorales. We wanted to see whether they are protective against mucormycosis caused by other mucorales. The antibodies are protective against rhizomucor, leucemia, mucorcelluloides, apophytomyces, as well as Cunningham-Miller. And there is, of course, variation in the way that you protect. And this is by itself, so keep that in mind. This is only antibodies, no antifungal being basically used here. So with that being said, we started looking into developing of monoclonal antibodies. So Lin and Sundus in the lab, they basically generated several hybridoma cells. These hybridoma cells basically generated antibodies that were as protective as the polyclonal antibody in the diabetic ketoacidosis mouse model. And then also C2 clone in particular was more protective than the rest of them in the neutropenic mouse model. And those antibodies, or C2 clone, was also protective against celluloides, mucor, as well as leucemia. So we're talking about 90% of all causes of mucormycosis are actually sensitive to this treatment. And I bet you if we've actually tried Cunningham-Miller, probably would see the same effect as well. Now, somebody asked about the antifungals with antibodies. Obviously, this is not meant to be a standalone therapy. This is going to be an adjunctive therapy with antifungals. And also what we wanted to do, we wanted to mimic the clinical situation. So mucormycosis, as you all know, is very hard to diagnose. So we wanted to infect mice and deliberately start the treatment 48 hours post-infection. This is really a delayed therapy, delayed established disease in the mouse. As you can see, that basically mortality starts really soon after infection. And what we did is we've treated either with posaconazole at 30 milligram per kilogram given twice daily for 7 days, or a single dose of the antibody at 30 microgram. Historically speaking, the posaconazole in this animal model does not really do very well. Sometimes it protects, sometimes it doesn't. But this is basically the kind of protection we get with antifungal therapy by itself. As you would expect, because of the delay in giving the antibodies, we have less protection. Instead of having the 70 to 80% and sometimes higher, we basically get 40%, which was statistically significant. But when we combined both, we lost not a single mouse. These are synergistic. We've tried this experiment as well with ambisome. We've gotten the same thing. I think approximately 80 to 90% survival. We've repeated it again with posaconazole. We've had the same effect. And also, we just recently did some CFUs as well. And the CFUs are approximately 2 log reduction in lungs and brains as well. So these antibodies are synergistic with antifungals. So just to summarize, passive immunization with the right antibodies protect mice from mucormycosis and seem to work. I don't think this is going to be a magic bullet that basically protect against all fungal infections. I mean, you really have to treat fungal infections as individuals, that they have specific characterization, and you really need to choose your target. What are you going for? So in our case here, COT-H seems to be a very important virulence factor that enables the organism to invade blood vessels and perhaps even lung cells as well. Mechanism of protection is reliant on prevention of invasion. COT-H also is ipsanophagocytic. It basically augments phagocytosis and also augments killing by superoxide and myeloperoxidase production. The most important thing is these antibodies are synergistic with antifungals, and they really make them look like dynamite in our animal models as well. So we are currently trying to humanize those antibodies and hopefully take them into manufacturing and eventually into clinical trials. So I would like to finish here by thanking people who have been instrumental in this work. In addition to the people that I showed you their pictures, I would like to specifically thank Yu Fu, who was instrumental in establishing the molecular manipulation of mucormycosis, which is not easy. It's really tough to manipulate this organism. Brad Spellberg initially working with me on this project, Jack and Scott for their mentorship and advice at all times. And last and definitely but not least is the funding from the NIH. This is all taxpayers' money, fully funded by NIH, and we haven't been able to attract any industrial interest in this because of the fact that mucormycosis is rare. And there has to be more reliance basically on governmental agencies to carry this forward. So I'll stop here and I'll be happy to answer any questions.